I'm sorry that this video is so long. To make it somewhat easier to watch, I've broken it down into 4 chapters. You can stop watching at the end of a chapter and resume watching whenever you have time. This video is long because the controversy I'm discussing has been raging on for more than 50 years. Sometimes people have taken their preferred theories and become combative with other electrologists. The issues I'm discussing are not settled yet. I'm hoping that an unabridged discussion of these ideas might offer new understanding to the simple question, what are we supposed to do to a follicle to cause permanent hair removal? Art Hinkle and St. Pierre proved as early as 1938 that hairs can be removed in all stages, anagen, catagen, and telogen. Hinkle's book was published in 1968. My book, The Blend Method, was published in 1991. Both books explain this oddly controversial fact. For a quick review, here are a couple of Hinkle's statements.
Now, if we want to stop any further generation of hair growth, we should therefore, it seems to me, destroy the side wall of the follicle that is going to contain the hair germs for the generation of the next follicle wall, as well as destroy the present papilla if it's an anagen hair, or if the hair is already going into catagen, the needle then will go down to the depth of a hair in anagen. We're putting our needle down at a point where we'll destroy the degenerating dermal papilla cells. What difference does it make whether we destroy them while they're young and tender or wait until they get in the degenerative stage? Whether we kill all the papilla cells while they're in their active stage or kill them while they're in the stage of being dormant, in which case they're still being kept alive by the outer connective tissue. This is why the whole theory is put forth in the book relative to making your insertions as deep as a hair in anagen. I figure if you go as deep as a hair in anagen, you have done everything you can to that area that's possible to do.
So why does the "you can only destroy a hair follicle in anagen" stubbornly persist, even in our schools? Most likely it's because of the plant metaphor that people assume is real. The first time you tweeze a hair, the thing you pull out, if you get the whole thing, undeniably looks like a root, the root of a plant. It's got a bulb like a plant. It's got a root like a plant. It's got a stalk like a plant. Indeed, those are the terms we use for hairs. Then, the first time you see an anatomical drawing of a hair, aha! You see the root ball being fed by blood vessels. Of course, people who know nothing about hair will tweeze a hair and think, well, if I pull out the root, the hair won't regrow again. Well, we all know that doesn't work. A hair is not a plant. A hair is not growing into the skin, it's growing out of the skin. But still, the plant metaphor persists in electrology. Here's the model. A plant dies if you cut off the nutrients to the plant. The plant metaphor in electrology argues that if you cut off the blood supply, the hair dies, much like a plant. Here's a photo from one of our leading electrology companies. Notice that they portray our goal as targeting the tiny capillary loop at the base of an anagen hair. Like a little sparkler, only the blood supply is being coagulated. Strangely, I've never seen a portrayal of the blood supply being coagulated in any growth phase other than anagen. Are we to assume that the blood supply disappears in catagen and telogen? Where did it go? Maybe this universal illustration gives us the idea that only hairs in anagen can be destroyed. However, this entire cut off the blood supply notion is the central concept that I'm going to challenge.
This is Eric, here to give you a break from Uncle Bono's raspy old voice. So, let's examine the rationale for the anagen-only protocol. Indeed, those who promote the practice are certainly not stupid, and we're not going to disparage the anagen-only practitioners. because, of course, they're getting permanent hair removal results. Indeed, a theory can be wrong, but still render a good outcome. Here's the way hairs in anagen are usually drawn. Here's the epidermis, and here's the dermis. The inner root sheath is colored white, as it appears when you epilate a hair. Here's the outer root sheath and, specifically, the bulge area of the outer root sheath. The outer root sheath is rich in stem cells. Now it gets interesting. Here's the dermal papilla. Notice that the proper name is dermal papilla. That's because the hair follicle's papilla is actually part of the dermis. The papilla is not part of the matrix of the hair. It's not part of the inner or outer root sheath. The papilla is not part of the follicle at all. The papilla is part of the dermis. The papilla is composed of specialized stem cells, connective tissues, and other components. Just above the hair papilla, I've drawn a layer of cells. These represent the hair matrix that, through cell mitosis, grows both the hair and the inner root sheath. The matrix is fed by the papilla. Specifically, the hair papilla is part of the connective tissue sheath, also called the dermal sheath. The connective tissue sheath envelops the follicle and underlies the entire epidermis, every inch of our skin. The connective tissue sheath is composed of connective tissues such as elastin, stem cells, nerves, blood vessels, and capillary loops. The blood vessels in the connective tissue sheath feed not only the hair, but also the epidermis as well. The connective tissue sheath is part of the dermis and is a blood-rich layer. At the base of the anagen hair, there's an opening in the papilla, where the capillary loop is implanted in the papilla. Notice that the papilla and the capillary loop are two distinct structures that work in harmony, blood vessels and stem cells. It's sometimes overlooked, but the papilla is not unique in the follicle only. That's why it's technically called the dermal papilla. Indeed, the entire upper layer of the dermis is called the papillary dermis because it contains millions of papillae that supply blood and nutrients to our hairs, the entire epidermis, and all other skinned appendages. I've drawn in a few red dots to show the location of the papillae that make up the millions of papillae that underlie the epidermis. That little upward pointing nipple is called the papilla, Latin for nipple. Each papilla contains a blood vessel, a capillary loop. Unfortunately, this is how an anagen hair is usually drawn, showing only a papilla at the base of the hair and identifying our target area as the papilla only.
Okay, old raspy voice is back. Here are a couple illustrations that will give you a better idea of the millions of papillae in the skin. Here are two papillae at the base of anagen follicles. And here are a few of the millions of papillae that underlie the entire epidermis. A papilla at the base of a hair and a papilla at the base of the epidermis are identical. They're the same structures. They look the same because they are the same, a papilla with a capillary loop. The entire papillary layer of the dermis supplies our skin and our hair with blood and vital nutrients. Here's another drawing that shows the upward-pointing papillae. I've drawn a capillary loop to show its location as part of the papilla. In some respects, the papilla and capillary should be called a papillary unit because it contains multiple elements. The upward-pointing nipple is the dermal papilla. The downward-pointing fingers of the epidermis that extend into the connective tissue sheath of the dermis are called reedy pegs or reedy ridges. These ridges create a greater surface area so the papilla and blood vessels can better supply nutrients to the bloodless epidermis. Here's a microscopic photo of the skin. Here you can see the upward-pointing papillae that are part of the papillary layer of the dermis and the connective tissue sheath. It's a little hard to see, but the papillae contain capillary loops. those tiny thread-like lines. The downward pointing fingers are the epidermal structures that envelop and surround the papillae. Again, these are called reedy pegs or reedy ridges. These bloodless epidermal reedy pegs absorb life-giving nutrients from the blood-rich papillae. This illustration is beautiful and my favorite. Here you see the abundant papillae that feed the epidermis. Notice too the plentiful blood vessels that populate the connective tissue sheath. These blood vessels support the papillae. Indeed, the connective tissue sheath is rich in blood vessels and other components. Now, just for fun, let's take this papilla and move it over here. Now, let's place an anagen hair on that papilla. I think you can see that whether a papilla is feeding the epidermis or feeding a hair and follicle, it's an identical structure. For some reason, our textbooks overlook this interesting detail.
Undeniably, there are hundreds of illustrations of hairs that are going through the shedding cycle. Most of them are pretty lame. This drawing, however, is from Dr. William Montagne's landmark textbook on skin and hair. Montaigne is still the key source of information for all of us, including most electrology writers. So let's look at it. Here, we see the anagen hair with a papilla. I suppose those little threads at the bottom represent the blood vessels that are within the papilla. It seems like it. Then poof, in catagen we see the papilla being held within the follicle. But where did the blood vessels go? Who ate them? Beamed up by space aliens? In telogen we see the papilla completely outside of the follicle, again with no blood vessels. Where did they go? The anagen-only group could rightly conclude from this drawing that in order to destroy the blood supply, the follicle has to be in anagen, because there are no blood vessels in catagen or telogen. This is seriously like, where's Waldo? I mean, where did the damn blood vessels go? Missing from this drawing, and it might give you a clue, is the blood-rich connective tissue sheath. I've drawn it in. Remember, this dermal sheath is rich in blood vessels, papillae, connective tissues, fibroblasts, etc., and is a true powerhouse for nourishing and healing the skin and hairs. Now, let's look at this beauty. The artist is showing the anagen hair connected to a blood supply. That's nice. However, that little bulb on the blood vessel sort of makes you think that that's the papilla. No, it's not. Then in catagen and telogen, the follicle shrinks up, but poof, where did the papilla go? This drawing seems to indicate the papilla stayed at the bottom. Really? This looks like Where's Waldo all over again, except this time, where did the damn papilla go? Seriously, is it any wonder there's confusion in our field?
Today, research on stem cells is uncovering new and amazing aspects of human biology. Nearly all our electrology textbooks are decades old and therefore not fully descriptive of the power of stem cells. Stem cell research is the new frontier in medicine. The following quotation is interesting. Hair dermal papilla cells are specialized mesenchymal cells that exist in the dermal papilla located at the bottom of hair follicles. Mesenchymal cells are capable of developing into connective tissues and blood vessels. This Montaigne drawing illustrates cells within the papilla and the connective tissue sheath. In this drawing, I believe Montaigne is showing fibroblast cells in the connective tissue sheath. I've colorized the papilla and blood-rich connective tissue sheath to show that the papilla and connective tissue sheath are essentially the same structure. Notice that a cell is migrating into the papilla. Montaigne's drawing does not include the capillary loop in the papilla, so I've drawn it in. Of course, the entire sheath is also alive with blood vessels. The connective tissue sheath is typically overlooked in our literature. It's much more than just a bunch of connective tissue. Indeed, this sheath is filled with important growth factors, specialized cells, and blood vessels. The literature presumes that papilla cells in the follicle's resting stage initiate new blood vessel growth when the dormant follicle decides to grow a brand new hair and follicle. In this way, when a new hair and follicle develops, it doesn't have to search around to find the original blood vessel. It grows a new one. How is that possible? Of course, we know that the papilla in Anagen is supplied with well-defined blood vessels. We also know that in catagen and telogen, the remnant papilla, now called a ball of dermal cells, contains no blood vessels. Consider also that the outer root sheath in all growth phases is packed with stem cells. Likewise, the papilla in all growth phases is comprised of specialized growth cells. The papilla and stem cells in catagen and telogen are sustained by the connective tissue sheath that is rich in blood vessels and growth factors. The papilla and follicle are not dead in catagen, in telogen, or in exogen. So if your electrolysis target is the papilla only, why does the hair have to be an anagen? The papilla is alive and well in catagen and telogen. Why not target the papilla in all growth phases? Indeed, we sometimes forget the awesome power of stem cells. Ostensibly, stem cells play the central role in all growth phases of hairs and follicles. This is a crummy illustration, but it shows how richly the follicle is supplied with blood and stem cells. Look at this. What is that? Well, that's you. We all started life as a single cell. Then the cells multiplied and developed into bones, skin, brains, eyes, and so forth. And the final product was you. If stem cells can grow a whole person, don't you think they'd be able to grow a new hair and follicle?
Okay, so to better visualize stem cell growth, let's talk about something we've all experienced. Yep, we've all experienced an abrasion, or two. Usually we'll think that we're going to be scarred from such an injury. However, unless the injury is severe, our skin heals perfectly. Let's see how this minor miracle happens. Here's a drawing of our skin. Here's the bloodless epidermis. Here are the reedy pegs of the epidermis that extend down into the connective tissue, or dermal, sheath. The basal layer of the epidermis and the reedy pegs are loaded with highly active stem cells. This is the underlying connective tissue sheath, also called the dermal sheath. This layer underlies the entire epidermis and envelops all hair follicles. This blood-rich layer also contains important healing factors. Here are the papillae, or the papillary units, that are part of the connective tissue sheath. These upward extending nipples contain specialized cells, growth elements, and capillaries. Below all these structures is the papillary dermis. This upper layer of the dermis is rich in blood vessels, essential cells, and other components. The connective tissue sheath is part of the papillary dermis. So, here you are making a beautiful, graceful 4-point landing from your bike. Your knees are bloody, which means that you have torn off a good portion of the epidermis right down to the papillary layer. Very quickly, a scab forms. Now, the stem cells in what's left of the epidermis migrate out under the scab. The stem cells, working with blood vessels, rebuild the entire epidermis. Yes, just a few remaining stem cells can rebuild the entire epidermis. The papillae, the reedy pegs, and all the layers of the epidermis. No scars, perfect healing. However, what happens if your abrasion removes the entire epidermis? Here's a hair follicle with a connective tissue sheath colored in. Your injury scrapes off the entire epidermis, all of it. You bleed and a thick, stiff scab forms. Amazingly, stem cells from the outer root sheath of the surrounding follicles, sebaceous glands, and eccrine glands migrate out onto the wounded skin, and once again rebuild the entire upper layers of the dermis and epidermis. We take them for granted, but stem cells are truly phenomenal.
So let me summarize so far. First, don't take drawings literally. Oftentimes we argue about hairs and follicles based on drawings. But remember, there are no illustrations that depict the actual follicle. All illustrations are artistic renderings, and most are incorrect. Here are a couple examples. In these two illustrations, the epidermis is shown too thick. Actually, most illustrations make this same mistake. These illustrations are artistic, but they're missing all the important parts. Drawings like these show nothing of any worth whatsoever. Drawings also give us the idea that blood vessels are permanent and unchanging, and that's not the case. Blood vessels are not static. Old, unnecessary blood vessels are absorbed, and new blood vessels grow as needed in a process called angiogenesis. Our bodies are dynamic and constantly changing. This is a schematic I drew a few years ago. Problem is, the sebaceous gland doesn't stick out like that. So here are actual hair follicles that were removed from a man's head. As you can see, it's nearly impossible to identify the various parts because, well, follicles aren't color-coded. Here are actual sebaceous glands, and as you can see, they look like little lumps of fat, not neat, well-defined structures. So the point is, don't get hung up on drawings. Don't base your arguments on drawings. Don't develop a technique based on drawings. Drawings lie, all of them.
Mike spent a lot of time talking about the papilla, and honestly, I'm sick of hearing about it. But still, the papilla is an important target for electrolysis, but it's not the only target. The papilla is not just a capillary, and it's not the only source of nutrients for the follicle. This is a capillary loop, but it's not yet a papilla. Now, with the surrounding dermal sheath, it is by definition a papilla, or a papillary unit. The papilla is part of the blood-rich dermal sheath and the papillary layer. The papilla inserts into the anagen hair bulb. The blood-rich dermal sheath envelops the entire follicle, and this layer feeds the hair and feeds the entire epidermis.
So I've watched several electrology videos that say once the hair bulb detaches from the papilla and goes into catagen, the follicle is dead. That's not true at all. Indeed, the regressing follicle goes through morphological changes, but it's not dead. If the follicle was actually dead, you could simply brush away the hair, or the hair would just fall out. Instead, as the follicle progresses to telogen, the hair and follicle are broken down, absorbed, and keratinized. If you ever pulled out or epilated a hair in telogen, you know how strongly it's attached to the upper skin. No, when the hair bulb detaches from the papilla, the follicle thrives and is kept alive by the surrounding blood-rich dermal sheath.
Mike also described how our skin heals after an injury. Even with most of the epidermis torn off, or all of it, stem cells in the skin are able to rebuild the skin. The vital cells of the epidermis are identical to the vital cells in the follicle. Cells in the epidermis regenerate damaged skin, and those identical cells regenerate new hairs.
Here's another illustration of what I'm highlighting, and this will be my last shot, I promise. For a closer look, I've enlarged this upper section and drawn it over here on the right. I've colored in the follicle and the epidermis to illustrate what all our teachers and textbooks say. The follicle is an indentation of the epidermis. And that means the follicle comprises all the cells and all the layers of the epidermis. Again, the dermal sheath nourishes the epidermis and follicle, in all stages. I've colored in the blood vessels to show that this layer is luxuriously supplied with blood vessels.
I hope you can see now that destroying only one blood vessel in the papilla at the base of an anagen hair is not going to cut off the blood supply to the follicle. There's so much going on in the follicle that simply anagen stage and papilla only is not enough. In fact, new research concludes that even if the papilla is destroyed, cells in the follicle eventually grow a new one. So yeah, you're going to have to do more than just zap the anagen papilla. But don't worry, you're probably doing it already. Now, let's move on and discuss the complete target area in electrolysis. The papilla, the follicle, and the stem cells.
This final chapter discusses how hair transplant techniques highlighted the incredible power of stem cells. To recap, for me, two relevant questions are settled. First, if the papilla is your target, then target the papilla in all stages, not just in anagen. Second, destroying the capillary at the bottom of an anagen hair is not going to cut off the blood supply to the whole follicle. I find it curious that Hinkle's book promoted targeting the papilla and the sidewalls of the follicle that contain the hair germ cells. As time went on, we focused more and more on the blood supply. It seems that we went from targeting follicle stem cells to targeting the surrounding blood vessels. To that point, the next intriguing notion is, can a follicle survive without a blood supply? And the apparent answer is, yes, it can. Indeed, the development of new hair restoration techniques point to interesting implications for electrolysis practitioners. Let's have a look. Dr. Robert Orentreich pioneered hair transplants as early as 1953. Dr. Orentreich was, by the way, a strong supporter of electrology. He was an outspoken and powerful ally during the years that we fought against the electronic tweezer scams. Back in the early years, physicians who were experimenting with hair grafts thought that, like skin grafts, transplanted follicles had to have an intact blood supply. They used an instrument called a skin punch. This tool works much like a little cookie cutter. It has a very sharp cutting edge. And with a downward and twisting motion, a core of skin is cut out and lifted out with forceps. When harvesting follicles, the goal was save the intact blood supply. The idea was not to disrupt blood vessels that feed the follicles. A large 4 to 5 millimeter diameter punch was inserted all the way down through the dermis and into the subdermis. A large plug of hair was then extracted. A same diameter hole was made where the hair plug would be then implanted. The donor site hole was left to fill in with scar tissue, and as you can see, the resulting scars were not acceptable. Not only were there scars in the donor site, but also the large hair plugs looked unnatural, like a doll's head. So for the next ensuing decades, surgeons experimented to create a more natural-looking head of hair.
Today, new procedures yield much better results than hair transplants of the early years. I'm going to briefly describe a couple procedures, but I don't want to make this a hair transplant video, because there's a ton of information on the internet. Instead, I want to focus on the blood supply issue, because hair transplant surgeons don't worry about maintaining the blood supply. Instead, follicles are harvested with no blood supply at all. And these new procedures work perfectly. Yeah, it's all about stem cells. Donor hairs can be harvested in several ways. This procedure harvests individual follicle units from the back of the head, using a small skin punch, or a power-assisted punch, or robotics. There are several trade names for such procedures, but it's often called an FUE, or follicle unit extraction procedure, or micro-mini grafting. The follicles are taken from the back of the head and implanted in the bald areas at the front. In this photo, notice that individual units of hairs are removed. As you can see, head hairs naturally grow in tiny clusters. Notice that there are no blood vessels removed, only follicle units. The surgeon harvests the entire follicle and the follicle stem cells. The donor holes are left to fill in and are nearly invisible. This procedure cuts out a strip of hairs from the back of the head. This is sometimes called the FUT, or follicular unit transplantation procedure. Here, the surgeon is cutting out the strip. I made this photo black and white because the color photo was just too gross. Here, you see the actual harvested strip of donor hair. If you look closely, you can see all the layers of the skin. That purple line is the surgeon's marker pin. From this strip of scalp, hair follicles are meticulously dissected into tiny hair and follicle grafts. Uncle Bono had this arduous task when he assisted Dr. Perkins in hair transplant surgery. This photo shows the simple cutting tools used: razor blade, tweezers, and a special cutting board. There is no scalp tissue in this photo. The donor site is sutured, and most of the time the scar is not noticeable. The tiny grafts are placed in saline solution or sometimes stacked in a ball of follicles. In this photo, I would assume the grafts would be cut into smaller units before implantation. Please look closely at these follicles. Notice that there are no blood vessels and no blood supply. Don't need it. You can see the whole follicle and some fat at the bottom. The fat is trimmed off before transplantation. Amazingly, these grafts can stay alive for many hours without a blood supply, as long as they are kept wet and don't dehydrate. Here we see the surgeon and an assistant implanting the grafts in the bald areas. The surgeon simply makes tiny punctures in the skin into which the graft is inserted. No hole is necessary. Those little purple lines are marks the surgeon made with his marker pen to locate where he'll make the slits. Indeed, all by themselves, without their own blood vessels or capillary loop in the papilla, the implanted follicles regrow new, vigorous hairs.
These are harvested grafts in Anagen. Take a close look at these grafts. Do you see any blood supply? Do you see any capillaries feeding a papilla? Well, you don't, because there's no blood supply at all, and the follicles didn't drop dead either. Incredibly, even when these little bundles of stem cells have been out of the body for hours, once implanted in the skin, they go to work and reestablish the entire network of blood vessels and follicles, and achieve their original depth and size. When I was assisting in transplant surgery, I was amazed that the follicles remained alive for hours before they were implanted in the skin. Often our surgery would take 5 hours or longer, and the harvested follicles were just fine. I asked 3 hair transplant surgeons if it's critical to save the papillae and the extracted follicles, and they all said probably not. Looking at these grafts, I can't tell if the papillae are still lodged in the bulbs. Maybe they are. Do you see anything? The surgeons didn't care as long as most of the stem cells were intact. I was also surprised because if there were not enough donor follicles, the surgeon cut the Anagen follicles in half and implanted both halves. They said each half would grow a new hair. I cut the grafts and implanted them too, but I never got used to the foul smell of human fat, really nasty stuff. During a different surgery, a chunk of fat fell on the floor and I slipped and fell on it. Yuck. I spent a lot of time fiddling with the harvested strips and follicles. I even inserted different electrology needles in the follicles. My experiments were very enlightening, but that's another story. I wish all electrologists had an opportunity to experiment with real human skin and follicles. A lot of our traditional beliefs would be overturned. And that's another story too. This is an amazing photo of dissected follicles, from anagen to telogen. Can you find the papilla? Notice that the papilla drifts up with the catagen and telogen follicle. And likewise, the papilla drifts down, or maybe it's pushed down, when the new anagen follicle developed. All the surgeons said that the telogen follicles are just as viable as the anagen follicles, and they had no problem using them at all. So I guess telogen follicles aren't dead after all.
Researchers have yet to discover all the populations of stem cells and how they function in the follicle. Lots of experiments are on mice. Claire Garcin and David Onzel discovered numerous sites of both stem cells and progenitor cells in mouse follicles. As their paper states, these cell populations might denote similar human stem cell populations. I've drawn in a red dot identifying all the stem cell populations. Clearly, follicles are packed with stem cells, and it appears that stem cells populate the entire follicle wall, not just the bulge, papilla, and hair germ areas. Again, transplant surgeons are pushing the boundaries in transplantation techniques as they discover new stem cell populations. In this next video, Dr. Cole describes how harvesting upper follicle stem cells can potentially deliver a new implanted follicle and preserve the original donor follicle, so you could get 2 hairs from 1 follicle.
The regeneration and the use of follicle stem cells for hair regeneration is an emerging trend predominantly in Europe. And in Europe, they are currently studying the administration of follicle stem cells back into the areas of hair loss or hair thinning with some interesting results. You know, we don't have any good studies yet, but I've seen some interesting clinical responses, some positive clinical responses. You know, follicle stem cells, when you think about them, there are different populations of follicle stem cells that are located at different regions on the hair follicle. For example, the CK15 positive stem cells are located approximately 0.8 millimeters to 1.6 millimeters below the surface of the skin along the hair follicle, and the CD34 follicle stem cells are located more at the bulb or the base of the follicle. So these follicle stem cells, different populations, lie along the follicle at different levels. And the reason for this is simple. When a punch enters the skin at an angle, or a needle enters the skin at an angle, the inferior bottom edge of the punch is going to incise deeper than the superior margin of the punch. And for this reason, we're able to leave some of the CK15 positive stem cells in the donor area. And so we leave this population of CK15 positive stem cells in the donor area, and then we basically pluck the hair follicle out. Plucking it out leaves the CD34 positive stem cells, and all the stem cells below the area of the incision are left in the donor area.
This video by Yale researchers looks at live mouse follicle stem cells using an innovative magnification technique.
To orient you a little bit, these are hair follicles. Here you see the hair shaft extending outside on the surface of the skin, and this is the structure that is rooted inside the dermis. The arrows are pointing to divisions, cell divisions that are taking place in real time. Are in fact regulated, which means certain cells are dividing in certain places and in certain orientations. Also, we found that cells within the hair follicle have the ability to reorganize and move directionally to support the organized growth of the hair follicle.
This is a very interesting experiment. These are follicle stem cells in a nutrient-rich solution. Without capillaries or a blood supply, the stem cells are developing into hair and follicle components. Perhaps this type of process is the future of hair transplantation.
So after watching this video, the question is, do I have to change my technique? I would say no, probably not at all. If you're getting good results, you're obviously doing it right, so don't worry about it. Of course, straight galvanic is famous for treating the whole follicle. However, the newer thermolysis machines, combined with the upgraded procedures for applying thermolysis, are definitely working well and treating the entire target area. Still, if you only want to treat anagen hairs, that's okay too. I will say that anagen hairs are much nicer to epilate. They just slide out. Not so much with telogen hairs. And of course, going anagen only, you're still going to get permanent results. In some instances, treating early anagen hairs makes sense. For example, if a person has distorted follicles, the early anagen follicles are straight, so it makes sense to zap them in the early stage. However, I question the strategy of anagen only and pre-shaving in all cases. For example, if you have a man with a hairy back, are you really going to have him shave his back and only allow a few days growth? If you follow such a protocol, you're going to extend the total treatment duration by months, even years. If you're doing an underarm case and follow the shave before treatment procedure, you're also going to significantly extend the treatment duration. If you have a woman shave her bikini area, you're going to encourage ingrown hairs. We're all taught to recommend women stop shaving and clip facial hairs. However, lots of women will not do it because they're paranoid of showing even the smallest clipped hairs. It's also nearly impossible to get some clients to shave. They're convinced that shaving makes hairs grow. However, shaving is better than plucking, so if you can get them to shave, not pluck, and present only a couple days' growth, well, that's definitely better than plucking. I find it remarkable that Art Hinkle's treat the papilla and the lower 2/3 of the follicle still holds true after all these decades. Whatever protocol you follow, and there are many different approaches, I hope that my video helps you create a more successful approach. Thanks, and please leave a comment.